Hello, everyone. Thank you for coming today. I'm sure you're all on the edge of your seats to learn more about nanoscience. Um, my name is Mishan, and here are my partners, Brandon and Christian. And today we're going to be talking about the varying levels of toxicity of different sizes of silver on yeast cells. So throughout this presentation, you'll learn that the smallest differences in the sizes of silver particles will have a significant effect on the yeast's health, which is measured by cellular respiration. For example, silver ions, the smallest type of particle in our experiment, are less than 1 nanometer in size. Silver nanoparticles, the medium-sized form of silver in our experiment, are somewhere between 20 to 50 nanometers, and bulk silver is in the micron range. So evidently, even the smallest differences in the sizes of silver have a very significant impact in experiments involving nanoscience. In fact, we're so accustomed to learning about these tiny, tiny particles that particles such as microns seem huge to us now. So the question is, why should you listen to us ramble on about microscopic silver particles? Well, silver is everywhere, and it's found in bandages, jewelry, circuit boards, water filters, etc. But this specific application of silver in nanoscience heavily contributes to important topics such as cancer research because it enables us to target specific destructive cells in the human body, and it allows us to kill them without doing any harm to the rest of the body. So to learn more about the process of killing these specific cells, I'm going to turn it over to Christian.
All right, guys. So, is this on? All right. So, as you can see up here, there are 3 different computer screens, and on them there are 3 different images which represent the various sizes of silver. So, on the far left, we have the nano-sized silver because, well, this type of silver actually causes the most damage on the cells, as you can see, like all the cracks. And in the middle, we have the bulk silver because it causes the least amount of damage. And on the far right, we have the silver ions, which cause like a medium level of damage. So the intruders and like the various houses that are like breaking in represent the silver particles in the experiment. And the house represents the cell because it's being attacked by the intruders, which represent the silver. And the various vehicles that you can see in these like little bubbles outside the house represent the sizes of the silver outside the cell. So basically, the nano-sized silver is shown to have like a bike that brings it to the cell, and it's like the medium-sized method of getting to the cell because the car is larger than the bike, and that's the bulk silver, and no transport is smaller than a bike, so it's the silver ions. And then you can see that the silver enters the house through various openings. So on the very left, you can see that the silver ions are entering through the door, which represents how the cells take in the nanosilver through a process of endocytosis. So basically, they mistake the silver nanoparticles as other objects, and the cell membrane kind of wraps around them and brings it into the cell, where then the ions can damage it. And then in the middle, you can see the bulk silver is like too big to enter the cell because like there's no openings or ways to, because the cell's so small and the bulk silver just can't get in. So what happens in fact is small ions break off outside of the large bulk silver particle, which is outside of the cell, and like very few of them can travel through special channels for small particles and do a slight amount of damage, which you can see like there's only one intruder damaging the house. And on the far right, the silver ions are entering through the door, and the reason why there's only like a small amount of cracks in the house is because the window is a lot smaller than the door, so they can only go in through like a slow rate. So the damage occurs more slowly. And the damage done to the house represents the level of toxicity that the silver has to the cells.
I think that this drawing did very well at showing the concepts that the artist decided to show. But like all drawings, we can't show everything. So there's one concept I'd like to point out, and it's how the nanosilver particles, which are represented by these on the left, when we used them in our experiment, they were yellow, and maybe we could have shown that by coloring each of the intruders in the house a different color. And I guess since the time is up, thank you for listening.
Any questions? Yes. Artificial intelligence to our nanoparticles. Possibly, if we want to further this science with cancer research, we can make nano-sized robots to transport these ions to these cells, to specific cells, to destroy these cancer cells. That's one way we can incorporate nano-sized silver. Yes. Yes, I think so because, yeast cells specifically undergo cellular respiration. So when all these different forms of silver get into the yeast cell, they basically catalyze the formation of reactive oxygen species, which is basically inhibiting their ability to consume oxygen, thus killing the cell. So I guess it could be dangerous if this reaches other cells and kills healthy cells.
Are we putting you in danger in this nanoscience camp?
No.
Correct. So why?
I believe that we learned in one of the lessons that the human body has like various vitamins that help protect it against the silver or prevent the cells from becoming damaged.
I think part of it is actually just that the silver particles don't get into the human cells the same way. But also it has a bunch of different enzymes than the yeast systems because it is a very different organism. But I think a lot of it is just that the silver particles are not eaten by human cells the same way they are eaten by yeast cells. So what would happen to you, here's a question, if you ate silver nanoparticles?
Well, I think we learned from one of the instructors that our body actually has antioxidants. So I think our cells would probably handle the silver nanoparticles better than how the yeast cells did.
Yeah, but something does happen to you if you eat large quantities of silver nanoparticles.
Oh, we would also turn blue.
Yes, you would turn bright blue. But otherwise would be very funny looking but relatively unharmed. Apparently. Don't try this at home. We're not condoning it. Okay, so my question is, as always, there's a limit to every analogy. So there's something fundamentally different about the nature of silver ions than about the nature of the nanoparticles. And what is that difference, and how might you reflect that in this picture? I mean, and it is actually sort of reflected, but how could you reflect it more?
Well,
share the mics around so that everybody gets to answer questions.
So I think what you're referring to is the different means of the different types of silver in getting into the cell. So, for example, if you're talking about bulk silver, it would have to dissociate outside of the cell, and all
it dissociates into what?
Silver ions.
Which have?
Charge.
Charge. How could you maybe reflect that?
Some sort of visual element showing that they're charged or something. set them apart?
I don't have a good answer. These are your opportunity to be creative. But the people all look sort of like the same people here. And in fact, neutrals and ions have very different interactions with things.
Right. So maybe that could be depicted in a difference of emotions between these people in the picture, which would show if they're positively charged or neutral.
Yeah, potentially. Anything about the looks could potentially be used to sort of depict that difference.